Peter, Sichin, and I will talk about how machine learning is accelerating the drug discovery process using AWS and the Machine Learning Solutions Lab. The structure of this talk will consist of 3 parts. The first part, we will give a broad introduction of what is the drug discovery cycle and give high-level examples of how machine learning is helping in this cycle. In the second one, we will dive deeper, and Sichin will give some details on one collaboration that we had with Janssen on one of these examples. And in the third part of the talk, Peter will be discussing how machine learning, AWS, and the Machine Learning Solutions Lab is helping Janssen to achieve their goals. So first of all, I think it's no secret that machine learning is helping and is slowly being adopted by the healthcare and life science industry. We have examples of how machine learning is helping to extract information from unstructured data, such as EHR or electronic health records, calls from telehealth, or claim notes from doctors. And another example is for clinical decision support systems, where machine learning is helping develop diagnostic systems that can help do screening or screen out protocols for clinicians, thus helping them reduce time and cost. And finally, there is the drug discovery development, in which we will give more examples of how machine learning is helping here. But first, let's start with the drug discovery or the drug development cycle. The process to get a chemical compound from the design to the pharmacy is very long and costly. So the first step here is the step where you have to ideate and design the way you want your compound to act on the target. It has to have the necessary properties and the necessary efficacy and specificity to be able to be viable as a drug. So once you have designed this compound, it needs to be tested in preclinical research using in vivo and in vitro studies, where you further refine whether your drug is viable for further steps in the process. After you have selected the compounds that have the highest success in these tests, you go to the clinical research part, and in this part, you have to test with humans, and you have to make sure that this compound is effective and also safe for human use. Thus, it can finally be approved, and once this is approved, it's not the end of the story. You can still, or you should still survey and audit your compound after it is approved to make sure that it keeps being effective and it keeps being safe. All of this process takes up to 10 years from candidate generation to approval and being on the post-marketing monitoring phase. And it is estimated that it has a cost of up to $1 billion to go through this entire process. So tools that help in this particular cycle are very, very important and very useful for the drug discovery. So now let's see how ML is participating in some of these steps in the cycle. And we will start with the drug discovery and development. For candidate generation, the task is to create new compounds. And the way you can do that are 2 ways. One of them is if you treat the compound in a simplified version as a SMILE, for instance, you can effectively translate this task to string generation. And for this, you can use different machine learning algorithms like autoencoders, recurrent neural networks, or even GANs. You can also do that taking into account the 3D structure, and for that you can use graph neural networks that model compounds as graphs of atoms and edges being covalent bonds. And we have seen several examples of those techniques being useful in this particular step. On the preclinical research, on those in vivo and in vitro studies that I was talking about, we can use machine learning to help us identify the drugs that are being harmful or not. In this particular example, the Machine Learning Solutions Lab had a collaboration with AstraZeneca to be able to screen images of tissues of the kidney and help clinicians identify which slides or which slices of tissue had damage. That also helped with identifying which drugs were being harmful to the particular subjects here. And in this particular sense, clinicians and machine learning work together in order to get those in vitro studies faster, to make them faster. The clinical research part, or the clinical trial part, is one of the most important ones because this part is one of the most expensive parts of the cycle. In this particular sense, machine learning has been trying to optimize basically how we can match patients with clinical trials. And the way we do that is by using EHR and natural language processing to find out of the protocols that are out there on the trials and the patients the pool of patients that we have there, which one are the best to match for each other. So we can do recommendations for clinical trials to particular patients and optimize the protocols and the site selection of this. And the final example that we will discuss about is, again, on the candidate screening. In this particular example, we can model the affinity between a protein and a chemical compound in a way that will allow us to predict, once we have more compounds, whether that compound will be affected to that target protein. We can also do a function prediction of the protein if we want to screen a bunch of candidates and we want to filter all of the candidates that are useful or particularly well suited for this particular task. And with that, I will leave my colleague Zichen explain and dive deep into this last use case that I was talking about.
Hi, thank you, Erica. My name is Zichen Wang. I'm going to dive deep into a particular use case that we collaboratively delivered to Janssen pharmaceutical companies. So as Erica mentioned, there are so many steps in the drug development process to bring a new drug to the market. And it requires multiple teams to work cooperatively to share data. And that's one of the key challenge that we faced in this proof of concept study that we worked together. So we will need to discover and harmonize and also connect the data sets across Janssen value chain. And to do that, we will need to build an intelligent data indexing system to connect the otherwise siloed data sets to create a system that will help multiple teams within the pharmaceutical company carrying out different functions so that they can work on the same information to create better drugs faster. So to start with this use case, our proposal is to develop a knowledge graph-based system. And this knowledge graph can be used to organize and connect those otherwise siloed data sets. And we also identified 4 key personas within Janssen who will use this knowledge graph. And those 4 personas are data scientists, IT engineers, business analysts, and batch scientists. So the data scientists will be able to query and create machine learning applications on top of those data sets, whereas the IT engineers will take care of the data ingestions and updating process. The business analyst will be able to create business intelligence reports on top of the knowledge graph organized data sets, whereas the batch scientist will primarily be querying the relevant data sets to a drug candidate of interest. So the next step of creating such a knowledge graph is to identify the key entities and their relationships among those entities. Here we identified two key subgraphs within the knowledge graph, which are assay graph, which connect molecules to their relevant experiments, as well as chemical properties from the laboratory notebooks and experimental results. And on the other hand, we also have a document subgraphs, which connect the drug candidate with their regulatory documents. So that from these slides, you can see all these different personas can use the knowledge graph to query the relevant information of the entity of their interest. So from an architectural perspective, this is how we build the Janssen knowledge graph. At the core of it, we have the data storage system that connect the relationships of the data and store various data types using appropriate databases. For example, we can have SQL for structured database and NoSQL for unstructured data. And we will use a graph database, Amazon Neptune, to store the data relationships. And on top of the data storage system, we can additionally build services on top of that to be able to query and also update the data sets within the data storage system. And on top of those APIs, we can build either machine learning applications, or it also can support very complex queries so that a scientist will be able to easily get a holistic view of, let's say, a drug candidate of interest. So next, I'm going to briefly talk about one of the case study that we built for Janssen, which is a machine learning system. We name it LM-GVP. It is used for predict molecular attributes for drug candidates, particularly protein-based drugs. This is a novel deep learning architecture that leverage both the protein sequence and structure information. So it does that by combining protein language model together with graph neural network. So on the protein language model side, it can learn contextual embedding from the amino acid residues from a protein. And these contextual embeddings are additionally used as node features within a graph neural network that's featurized from a protein 3D structure. And this is a end-to-end ML learning system that we can fully backpropagate the gradients to predict the protein's attributes or functions. Our results show that it has achieved the state-of-the-art performance on multiple protein function prediction datasets. And furthermore, it also provides residue-level interpretability. So here is an example demonstrating a particular class of protein candidates that are able to carry out the function of ATP binding. By highlighting the important residues that's responsible for this function, we not only find that it recovers a known ATP binding motif, but we also found that those residues are structurally close, so they are located at a particular active site on multiple of these protein candidates. Highlighting this architecture can not only interpret and predict the function based on sequence, but also based on protein structure. So next, I'm going to hand over to Dr. Peter Clark, to talk more about the institutional, how he leveraged institutional leverage and active learning to accelerate pharmaceutical research and development.
Thank you, Zichen. It's my pleasure today to kind of extrapolate and build upon these core tenets and concepts, and hopefully further kind of elaborate on how we're really making these real and applying these to practice within the pharmaceutical industry. And so, my name is Peter Clark. I'm the head of computational science and engineering within Janssen R&D within the Therapeutics Discovery Organization, and we're fundamentally responsible with taking drugs from target all the way through NME. And so, as Erica had noted earlier, this is a very long and expensive process, and we're effectively leveraging various components of digital innovation, both on the platforms and capability side, to really accelerate therapeutics discovery and bring better and differentiated molecules to the clinic faster to treat patients with large unmet needs. And so this, of course, kind of spans the pre-portfolio space all the way into preclinical development, clinical development, and late-stage product life cycle. And what we're doing is effectively leveraging data across the product value chain as incorporated into the knowledge graph to really inform molecular design optimization earlier in the R&D product life cycle. And so what this looks like is, you can imagine, you have various failure modes and attrition across various stages of clinical development. And you can, of course, learn from these mistakes and these shortcomings. And so this includes reverse translation of manufacturing data, large molecule CMC and API data, as well as clinical trial data to better inform not only trial design, but also molecular design optimization of these molecules. And so foundational to this really is that underlying data structure and architecture, that underlying data model. And to Zichen's point, we've developed this essentially knowledge graph, an R&D knowledge graph. And so this allows you to really unify these disparate data, as well as go back and bring in legacy data as well as prospective data into the knowledge graph to really inform these models in real time. And so, here we're capturing data from all across the organization and the product lifecycle and value chain around specific methods, the molecules, the formulation, the manufacturing process, analytical development, and so on and so forth. And really leveraging these data, first of all, to provide access to these data for scientists so they can fundamentally ask questions and have data that's discoverable and interpretable, but also really providing a ground source of truth on which scientists can operate and inform their learnings downstream. So we basically executed an early stage POC where we demonstrated a lot of these benefits to the organization. Fundamentally just around having a unified data source, the ability to track assets all the way from target discovery and validation all the way through large molecule manufacturing, API, and into the clinic. It's scalable and, of course, readily can be adjusted to ingest different types of data or new data streams, whether it be longitudinal data, various assay data, and/or metadata around a molecule or set of molecules or programs. And what this really enables is, of course, users from all different types of functions and business functions across the organization to operate on this kind of basic ground truth source of data to inform learnings. And so this can look like, what's the next best experiment to perform? Are there any existing data gaps around a specific portfolio program or set of molecules? And kind of how we can best accelerate on a therapeutic program going into the clinic. So this, of course, includes the legacy curation of legacy data to basically feed into the graph and kind of develop that institutional knowledge, that shared institutional knowledge. And so this was done primarily using NLP-based approaches to really go into semi-structured or unstructured data assets and make those findings visible, infer some meaning from those data, and then publish them into the graph so that these data aren't effectively lost and can be leveraged for future programs or future scientific endeavors. So to give you kind of a case of how, specifically on how the machine learning techniques are really advancing our scientific work and accelerating the product life cycle here is on the antibody side and protein therapeutics in general, we essentially rely on high throughput laboratory techniques and in vivo antibody generation to produce on the scale of thousands of unique, or tens of thousands even, of potentially therapeutic antibodies. And so screening these molecules from a functional perspective becomes effectively intractable. And so these in silico models really provide us with a tool to be able to predict a lot of these endpoint assays for things like developability, as well as, to a certain extent, PK/PD characteristics of a molecule, and really prioritize them from the sequence and structure alone very early in the portfolio, such that these molecules fail essentially at the onset. And so the LM-GVP model in particular, leveraging both antibody sequence and structure from those earliest stages of characterization, really allows us to effectively prioritize and triage leads based on several predicted endpoints. So paratope prediction, PTM prediction, or post-translational modification prediction, thermostability prediction, immunogenicity prediction, and in silico developability assessments. And then, of course, the idea here is that these models, informed by or basically an abstraction or kind of furthering the efforts of the initial LM-GVP work for specific tasks, and tuning those models for these specific tasks to deliver those results to wet bench scientists to make the best decision possible when it comes to advancing therapeutic leads. So the other side of the coin here is, of course, not only leveraging the institutional data in the knowledge graph and these models in an active learning context to iteratively refine and update those models, but also to make these the output of these models available to the scientists. And so we've spent a tremendous amount of effort also kind of building out a lot of the UX, UI capabilities in order to kind of deliver these models in a meaningful way where they can be interpreted by scientists in the context of the sequence and the structure of the molecule and related molecules. So to dig in a little bit further on that piece, we're using the LM-GVP model, as Zichen said, which is informed by 2.1 billion protein sequences and 393 billion amino acids to really fundamentally understand what it means to be a protein. And leveraging this language model together with the geometric vector perceptron, which essentially is a 3D representation of a molecule in space, the atoms in space, to better inform and really boost the performance of the traditional language model approaches. And so what we found is that this specific architecture and model is particularly powerful at determining functional residues of molecules. And so you can leverage the beauty of the language model, right? Is you can really leverage these the attention weights or the saliency maps of these models to inform the deterministic residues for any particular task. And so this really is pretty powerful in terms of really understanding the fitness landscape of a protein and how you would kind of best perturb a molecule towards a specific endpoint or range of values of interest. So to take it one step further, prediction is one thing. What I think the aim of the field really is to do is to apply these predictive models as discriminators in against your generative function. And so the holy grail of protein and peptide therapeutics is really kind of this ab initio approach, where you start with a target structure, you put it into a computer, and you say, spit me out a drug. And I think, of course, we're certainly a ways away from that in many regards. But through significant advancements in the field, both through empirical resolution of native physiologic structure of protein targets through techniques like cryo-electron microscopy, and in silico approaches like AlphaFold, really give us a much better starting point for this problem. And so, kind of digging in on this, right, it's not just enough to be able to have a Monte Carlo simulation or generate a deep fake of an antibody and expect it to have any amount of therapeutic properties. And so starting with that structure, you effectively need to be able to design a molecule that, that packs with the target protein or has an optimized protein-protein interface between your therapeutic molecule and the desired target of interest. And so we effectively sought to tackle this problem using quantum computing because it's essentially an NP-hard problem. And so you can think of a protein in the way it folds basically as these lower order and higher order interacting terms. And effectively, as you grow a protein or amino acid chain or fundamentally a string of amino acids, you have basically an exponential increase in the number of resulting molecules. You have essentially 60 unique rotamers or atomic configurations of a protein, of amino acid, and as you grow that chain, it effectively becomes intractable to solve using conventional computing. And so we sought to formulate this problem as a quantum computing problem, specifically as an optimization problem, which is amenable towards solvers such as the quantum annealing, such as D-Wave, as well as gated computing-based approaches. And so what we did was we effectively formulated the problem in twofold. The first being a 4 amino acid peptide design problem and a 14 amino acid peptide design problem. And we did this for two reasons. So the first is that we wanted to really understand what the empirical guardrails are, kind of how big of a problem can you embed on current day quantum computing? What are the limitations of that hardware? And then a real world example, right? So if we want to design larger proteins, we have to scale this problem. And so we evaluated both problems in terms of not only the goodness of fit of that solution, but also the compute time required for that solution. So towards this end, we essentially relied on a hybrid quantum and standard canonical compute architecture. And we formulated the problem, especially for the former problem, or 4 amino acid peptide problem, we're able to embed fully on D-Wave, that fully connected graph. But as you grow the problem, of course, you can't fully embed that problem onto D-Wave. And so we relied on a QUBO formulation of the problem to effectively rely on both classic as well as quantum compute. It's a QUBO formulation to essentially solve larger problems. And so quickly reviewing kind of the overall compute and the fitness of solution here, you can see that on the 4 amino acid problem, which you could fully embed on D-Wave, the fitness of solution is the same. The amino acid sequence here is exactly the same for each solver. Of course, the compute time is quite variable, where with Bracket having a significant time, with the D-Wave platform as implemented on Bracket having a significant advantage in terms of the compute time as compared to standard numerical solvers or classical approaches. We also included the QB-solve approach here to kind of apples-to-apples comparison between the 4-mer and the 14-mer amino acid design problem. And you can see that for designing larger proteins, optimizing the packing interface of larger proteins, you effectively you can embed this directly on the D-Wave platform. So we're left with the QB-solve formulation of the problem, which converged in 3.5 hours and by far gave the best fitness of solution as compared to the classical solvers FICO Express and Taboo, which in the case of FICO never actually converged on a solution in any reasonable amount of time. And the Taboo solver, which took 1,000 seconds, but again found that local optimum. And so this kind of demonstrates the application of quantum computing, in particular, to move beyond the generative model space beyond the predictive modeling space into the generative modeling space, and really advance these efforts across industry. So with that, I just want to thank Erica and Zichen for their help on these projects, and working with the team has been fantastic. And kind of, we're leveraging these various digital innovation tools and platforms, from the knowledge graph through machine learning and artificial intelligence models and reinforcement learning, as well as quantum computing, to really accelerate and deliver on that promise of better medicines faster for our patients. Thank you.